Hi, my name is Laura. I'm a postdoctoral researcher at the University of Toronto. So I'm working in the interface of synthetic biology and stem cell bioengineering. So I work with human stem cells that have the ability to self-replicate and also to produce defined cell types in vitro in the dish in a lab. So what I aim to do is to develop synthetic gene networks that are basically a combination of genes that do not yet exist in human stem cells, introduce those into the cells and to perform a specific task. So one of the possible tasks I want to implement is to detect specific cell types of differentiated cells, why we differentiate stem cells. Because we basically don't understand very well how the in vitro differentiation of stem cells happens, and it is often case inefficient. If we can solve the problem, we can potentially in the future produce cell types for therapies out of stem cells. Yes, so we are using two CRISPR-Cas in our lab, and it has obviously a lot of potential. But I have to say also in the past, genome editing tools have been developed and used, so they exist for a while. Just CRISPR-Cas is like a much easier way how to engineer it and how also to multiplex it, so you could in theory manipulate multiple genes at the same time. At the same time, for stem cells, or in generally, what works very well is, for example, to knock out the gene, and we call, or it is often called precise genome editing, but in reality what happens is that you produce random mutation at a precise location, right? After coupling of the Cas9 on the genome, the repair mechanisms of the cells to close the cutted site is really imprecise, right? So we introduce nucleotides or we delete nucleotides, and it's often case unpredictable, right? There is also a way how to use CRISPR-Cas in a more precise way by introducing an external DNA fragment, and in this way you could really do precise genome editing, but this process is dependent on the cell's ability to perform homologous recombination, and the ability of, or the rate of homologous recombination is, for example, in stem cells really, really low. That makes the whole process really, really inefficient, right? So there is a lot of room of improvement, but I have to say also just within the past few years there have been so many more strategies on CRISPR-Cas9 and how we could improve that. So it's progressing very fast. So in January, from a research or scientific perspective, I don't think there should be any limitation. At this point it's really early, and we're just starting to explore that. And I think what is important also to harness this potential to make progress in understanding really basic stuff in biology, and that's, I guess, in first place the most potential of the tool at this point, right? In future, or not even in future, already now the tool is used for therapeutic approach, and there are already clinical trials ongoing. We have a lot of clinical trials now starting that involves the genome editing of immune cells, or using genetically modified immune cells, or we have clinical trials in genome edited hematopoietic progenitor cells for the treatment of HIV. There is many other examples, and I think those trials will expand a lot in future, and time will tell us how well this works. At the same time, we obviously have to improve the tools itself and our understanding. Recently there has been this paper where they genetically modified human embryos, right? So from a scientific perspective, again, everything that you achieve can be of knowledge and you will learn from it, right? And sometimes it's just to show that you can do it and you will learn from it. As I mentioned, I should maybe not set limitations yet, but then when it of course goes towards applications or therapy, we need to discuss that, and it has of course to be part of our society and culture and ethical interpretation or interests, and eventually even if it gets approved and ethically accepted, it will be a really private decision to do that, right? As even now in some countries you can do in vitro fertilization, right? Many wouldn't do that, right? In future, maybe you can imagine to first genetically modify your germ layers instead of taking a healthy sperm from a donor, right? Eventually, it would lead to the same product, but it is a really private decision to go to that point. Also, in future, assuming the technology is very safe, right? So what you do if you have a child and the child doesn't see well, you give him glasses, right? And if in future you can do the same by editing his cells without any risk, right? And you improve his vision. So would you do it or not? And if it costs less than the glasses, would you do it or not, right? So again, I guess if it has proven that it's safe and accepted, then it's individual's decision if he wants to do that or not, right? So it's difficult, right? I think science communication in general is very difficult, and there should be a lot of effort in it coming not from just individual persons, but from university, schools, and in the first place, I think people need to get educated and understand a bit what is biology and take the fear from them of certain terms, right? People are afraid of terms like genes, terms like embryos, terms like viruses, and they sound frightening, but sometimes it's part of our lives. And I think if they would understand just a little bit more, it would be easier also to take the fear from many technologies that are currently under development. At the same time, of course, we have to communicate correctly also what is the potential, but the potential is, of course, great. It can really impact our future life, and that's why I think we should start in the core of education and communication of really the basics. Yeah, so I definitely think or have the hope that gene-modified cells or gene therapy will change our lives in terms of a therapeutic perspective, and that we can cure disease that we haven't been able to cure, and at the same time maybe to think of a way that it also gets more cost effective, right? So if you think of cells as a therapeutic, it might stay costly for a while, I guess, but it depends for each case or each disease, right? But if you would compare treatment of a complex disease, if you could do in vivo editing, all what you need material is a virus or a construct that has Cas9 on it, right? And if it works, you could administer that. It's actually a cheap way how to treat diseases. If you think again of engineered cells, that is more costly. But if you compare it to other treatments today that are combined approaches, of course, very difficult and time-consuming technologies, I think it has the potential to reduce the cost, but it will initially, of course, not be cheap. But it has the potential. So apart from the healthcare sector, CRISPR-Cas genome editing tools have, of course, also impact in other areas in our life, and I think what would come up in the next 10 years is a lot of in the food and agriculture industry. This also depends heavily on how the public will accept the technology, right? But this could improve our life, reduce pesticides, reduce the amount of water we use. But there has to be done a lot of improvement, and especially also in the communication. I think in the past there has been maybe bad reputation and companies misusing or miscommunicating those technologies, right? But I think the next generation of those scientists and entrepreneurs, they have, or I hope they have, a really good spirit and they want to use those technology for the right reasons, and that's really important, right? And that's what should be communicated to the public. This will help a lot to increase the awareness and power of